This is an AQA Trilogy paper, Biology 1H from May 2020, GCSE. A student investigated the effect of different concentrations of sugar solution on pieces of potato. This is the method used. Cut 5 pieces of potato, record the starting mass, place each piece of potato in a different concentration of sugar solution. After 24 hours, remove the pieces of potato, record the final mass, and calculate the change in mass. What is the independent variable? Remember, that's the one that you change. So what did they change about it? Well, it was the concentration of sugar solution. Table 1 shows the results. Explain why the potato in 0.0 molar sugar solution increased in mass. It's all to do with osmosis. It's because there's more water in the surrounding solution. And how does water move? By osmosis. So why does the potato increase in mass? Because water enters the potato by osmosis from an area. Remember your definition of osmosis. So from an area of high water concentration to an area of low water concentration, and just point out where those things are. The low water concentrations, in this case, inside the potato. Complete figure 1. Some of the results have been plotted for you. You should plot the data from table 1 and draw a line of best fit. So we have change in mass on the y-axis together with concentration of sugar solution on the x-axis, and they've done 0.3, 4 and 5 for us. So we're looking at these results here. So at zero concentration, it's 2.2. At 0.1, it's 1.15. At 0.2, it's 0.25. Drawing a line of best fit through the points, so a nice smooth curve here. Remember, with the line of best fit, you're looking for as near to an even distribution of points on either side of the line. Slightly off here, but I'm struggling because of the iPad. The mass of a piece of potato does not change when the concentration of solution inside the cells is the concentration of solution outside the cells. Determine the concentration of sugar solution inside the potato cells. So by definition, that's where the change in mass is 0 grams. So we're looking for that point here. So what is the concentration? We'll make sure you read off. That's 0.2, 0.3, and that will vary slightly based on how you've drawn your line of best fit. So to 2 significant figures, that's 0.22. Table 1 is repeated below. Calculate the percentage change in mass for the potato in 0.2 molars sugar solution. And look, they've given us the equation. So making sure that we give our answer to 3 significant figures. What is the change in mass? Well, we've been told it's 0.25. Write out that equation again so you don't introduce any small errors. So we know it was 0.25. Well, what is the mass of the potato to begin with? 7.96. And then multiply it by 100 to get 3.14070. And we want our answer to 3 significant figures, so count the number 1, 2, 3. That is your final answer. Starch is digested to form sugar molecules in the digestive system. What is the name of the enzyme that digests starch? It's amylase. Remember to learn what amylase, protease and lipase will do. Where are most food molecules absorbed? So where has a very large surface area to aid the absorption of food molecules? Well, that's in the small intestine. Remember, the large intestine absorbs water, liver produces bile, and the stomach produces protease and acid. Figure 2 shows 2 villi. Figure 2 also shows one cell on the surface of a villus is seen using an electron microscope. So here's that large surface area I was talking about, and then we've got some microvilli on the surface, further increasing the surface area more. You have one advantage of using an electron microscope compared with using a light microscope. Well, you can see more clearly, and remember the way we write that is higher resolution with electron microscope. You could have also written possible to achieve greater magnification. What type of blood vessel is labelled X? The teeny tiny blood vessels you find inside a villus is known as a capillary. Remember, arteries and veins are far too big to be found inside an individual villus. The real length of one villus is 0.8 millimetres. Calculate the image length if the villus is viewed at a magnification of times 20 and use this equation. So we know the magnification is 20 times 20, so we put 20 there. We're being asked to find the size of the image, so I'm going to pop an X there. And then what is the size of the real object? Well, that's 0.8 according to the question. Check our units. Our answer also needs to be in millimetres. So how do we solve this for X? By multiplying both sides by 0.8 to get 16 millimetres as our final answer. Figure 3 shows two cells from the surface of a villus. There are sugar molecules inside and next to each cell. So there's our sugar molecule. Name the process by which sugar moves into cell A. So here the sugar molecules are at high concentration. They're a much lower concentration over here. So we know it's going to occur in that direction due to the fact that we're told it's moved into the cell from an area of high concentration to an area of low concentration, which is why it's diffusion. Name the process by which sugar moves into cell B. This time we're looking at an area of low concentration moving to an area of high concentration. Remember, this requires energy. It's active transport. Give one use of sugar in the body. The easiest answer here is as an energy source. You could have also said it's used in respiration or to form glycogen. I prefer energy source. Figure 2 is repeated below. Explain how villi are adapted for efficient absorption of sugar molecules. So what can we see here? Let's see what we see. Right, let's point it out. Microvilli and villi increase the surface area due to those infoldings. We've also got a mention of mitochondria, so don't ignore those. Many mitochondria. Well, what do mitochondria do? They carry out respiration. And why is that important? To release energy. And why might you need to release energy? Well, some of the absorption occurs by active transport. Where do those food molecules go? Well, they go into vessel X, which remember is a capillary. So we're going to say, look, we've got lots of blood capillaries. We can also mention, I know it's hard to see, but the villus wall is very thin, which provides a short diffusion distance. Being overweight can affect the health and life expectancy of a person. Give one disease related to being overweight. Lots of options here. I'm going to put diabetes. You could have also put coronary heart disease, cancer, depression, high blood pressure. The BMI body mass index helps to show if a person has a healthy body mass for their height. It's calculated using this equation. A woman has a BMI of 27 and a body mass of 68.1 kilograms. Calculate the woman's height. So, in order to get this right, we're going to accurately write out that equation. Remembering that we need to square the height. So the BMI we've been told is 27. The body mass is 68.1. And we're after x squared. Remember, in order to solve for x squared, you have to multiply both sides by x squared. So it becomes 68.1 divided by 27 equals x squared. And that's 2.52 recurring. And then remember to square root to find x. to get 1.588. So to 3 significant figures, 1.59. Figure 4 shows a height body mass chart for adults. Which weight category describes the woman in question 3.2? So we know that their height was 1.59 and that their body mass was 68.1. So we're looking along here and up here, just approximation. So draw a line up from both points and look at where they cross. It occurs in here. So we know that this person would be counted as overweight. People are encouraged to control their body mass with diet and exercise. Describe how the balance between the mass of food eaten and the amount of exercise a person does controls body mass. So the key thing here is to look at the mass of food eaten and how that would affect body mass, and then the amount of exercise done, and then how that affects body mass. So don't stress too much about that, because obviously if you eat more food, you're going to increase your energy intake. And if that energy intake is greater than the energy transferred by the body, then you will gain mass. If you exercise more, then you transfer more energy. And lastly, if energy intake equals energy transfer, your body mass stays the same. You can write anything here though, honestly. During long periods of vigorous exercise, the body respires anaerobically. Explain the changes that happen in the body during and after vigorous exercise. Okay, so what happens when you exercise? When you exercise, your breathing rate increases. Why? To get more oxygen into your body. And why is that oxygen needed? For aerobic respiration. What else increases? Heart rate increases to deliver that oxygen to respiring cells so that they can release energy to contract muscles needed to exercise. However, if your exercise is too vigorous, you run out of oxygen. So in intense exercise, anaerobic respiration takes place. Let's write the equation for that. Remember, it takes glucose and it breaks it down into lactic acid. It releases much less energy. Remember that an oxygen debt is created. And so if we consider the wording of the question, what happens after exercise? Well, our breathing rate stays high. And why is that? To repay our oxygen debt. So our breathing rate stays high to remove lactic acid, which would otherwise cause muscle fatigue because it's toxic. There are tons of marking points available here. So let's highlight the marking points. Breathing rate increases. More oxygen needed for aerobic respiration is the second mark. Heart rate increases, third mark. To deliver oxygen to respiring cells is a fourth mark. And then in intense exercise, anaerobic respiration takes place, which produces lactic acid as a mark. And then the oxygen debt needing to be paid back by the breathing rate staying high is also a mark, in order to remove lactic acid and to prevent muscle fatigue. Cells are the basic units of all forms of life. Describe 4 differences between a bacterial cell and a plant cell. Let's name some structural differences. So here's a bacterial cell. Has a cell wall, has a nucleoid rather than a nucleus, and it has small circles of DNA known as plasmids. It has a tail called a flagellum. Our plant cell, remember, is kind of this shaped. It has a vacuole in the middle, a nucleus, chloroplasts, ribosomes, which actually the bacterial cell also has. So what's the main difference here? Well, bacterial cell, let's write a full comparison, has a nucleoid or a circular chromosome. Plant cell has a cell wall. Plant cell has chloroplasts, so it can carry out photosynthesis. Bacterial cell does not. The bacterial cell has that flagellum, that tail to help it move. The plant cell does not. Bacterial cell has plasmids. I'm so sorry, there's a storm and a bird. I would never make a video if I waited for there to not be a single sound. You could have also talked about things like the bacterial cell is smaller than the plant cell. The bacterial cell doesn't have mitochondria, the plant cell does. The cell wall of a bacterium is not made out of cellulose. Gonorrhea is a bacterial disease. A new vaccine is being developed against gonorrhea. Describe how a vaccine would work to prevent gonorrhea. So, a vaccine contains a dead or weakened form of the pathogen, gonorrhea. So what happens then? Lymphocytes are the white blood cells that are important here. They recognize the antigen, which remember is the special protein marker on the pathogen that identifies it specifically, in this case as being gonorrhea. And how do lymphocytes work? They produce antibodies against the pathogen. But what about this whole long-term protection? Well, it's because some lymphocytes turn into memory cells, which means upon reinfection with that gonorrhea pathogen, the crucial thing here is that antibodies are made. 3 words are important here: sooner, faster, and in greater quantity. And that's what stops you getting sick from the gonorrhea, because those memory cells are already in place to produce large numbers of antibodies. Another disease caused by bacteria is salmonella food poisoning. In the UK, chickens are vaccinated against the salmonella to reduce the number of cases of food poisoning in humans. Explain how vaccinating chickens reduces the number of cases of salmonella food poisoning. So, vaccinating chickens means they're not infected with salmonella. And so from a human point of view, they don't consume the salmonella, meaning they can't get food poisoning. Give one way that the spread of salmonella food poisoning from one human to another is controlled. Do not include vaccination in your answer. Let's just wash our hands when preparing that raw chicken. We should always be washing our hands. If anything, COVID has taught us that. You could have also said wash your hands after using the toilet. The number of cases of salmonella food poisoning is usually higher in summer than the winter. Why? Okay, well, what's crucial about summer? It's hotter. And so those bacteria replicate faster. Why? Because the enzymes work more quickly, because they have greater kinetic energy. This question is about photosynthesis and food production. How can oxygen production be used to show the rate of photosynthesis? Okay, well, oxygen is a byproduct of photosynthesis. Remember, the equation is carbon dioxide plus water forms glucose and oxygen. and a faster rate of oxygen production indicates a high rate of photosynthesis. Scientists investigate factors which affect the rate of photosynthesis in tomato plants. The tomato plants were grown in a commercial greenhouse in the UK during winter. Here's our light intensity. We've got carbon dioxide levels on the right and a rate of oxygen production on the left. The percentage of carbon dioxide in the Earth's atmosphere is 0.04%. Name the factor that is limiting the rate of photosynthesis at point X. Well, what can we see is changing along here? We can see that for this dotted line, the carbon dioxide remains the same, so it can't be that. It's this light intensity is increasing along the x-axis, so that's why it's light intensity. Farmers growing tomatoes commercially try to control the rate of photosynthesis to make maximum profit. A farmer can control the temperature and carbon dioxide concentration in a greenhouse. What is the minimum light intensity a farmer should use to get the maximum rate of photosynthesis, shown in figure 5? So you're looking for the maximum oxygen production before it starts plateauing, and it occurs here. So let's read down on the x-axis, and it's 20,000 lux. The light intensity you gave in question 5.3 may not give the farmer maximum profit. Explain why. So if you look at this graph, it's not just light intensity which is affecting the rate of oxygen production, i.e. the rate of photosynthesis. We also need to consider temperature. We can see at higher temperatures, photosynthesis occurs faster, as well as at higher carbon dioxide levels, and all of these things cost money, because look, this is atmospheric CO2 here. So why might that light intensity not give the farmer the maximum profits? There are other costs, such as heating the greenhouse and increasing the concentration of carbon dioxide in the air. And so these costs might be greater than the additional sale cost generated by the tomatoes. Explain the results when the light intensity was 0 lux. Wow, very little photosynthesis taking place, and in fact you're seeing oxygen actually being used up here because we've reached negative levels. So remember, what process requires oxygen? It's respiration. So we also need to talk about that in our answer. So let's write a scientific explanation. At no light intensity, no photosynthesis takes place, meaning that no oxygen is produced. The tomatoes actually use up oxygen in their respiration, meaning that there is a net loss in oxygen overall. New drugs are tested and trialled before they can be licensed to treat patients. Figure 6 shows how much time the different stages of testing took place for one new drug. So this is the preclinical testing that takes place on either animal cells or tissues. These are the healthy volunteers, these are the ones suffering, and these are the ones where we're considering dosage. How much more time did the clinical trials take compared to the preclinical testing? Okay, so let's work out how long that took. And then let's change colour, and we need to add these ones all together because this is all clinical. So for preclinical testing, we can see here it took 3.5 years. And then underneath we've got 1 year, 1.5 years, 4 years. So in total, 6.5 years. For clinical testing compared with 3.5 years for preclinical testing, so the difference between that is 3 years. During phase 1 clinical trial, low doses of the drug were tested on healthy volunteers. Suggests one reason why low doses of the drug are used in phase 1 clinical trials. Well, you just don't know how toxic they're going to be or if there's going to be any side effects. Suggests 2 reasons why healthy volunteers are used in phase 1 clinical trials. Well, that's because if there's a risk of this drug being toxic, you can't be using it on people who are already unwell because it's quite likely to be very serious for them. So testing drugs on ill patients poses too much of a risk for them. You can argue it the other way. Testing drugs on well patients or healthy volunteers poses little risk to them. And the other thing is, it's easier to identify side effects on people who are already well or healthy. The results of clinical trials can only be published after peer review by other scientists. Suggests one reasons why the results must be reviewed by other scientists. Well, it's to basically stop false claims of how good your drug is. A drug is only licensed for the medical conditions it was tested to treat in the clinical trials. Drug regulations control what drugs a doctor can prescribe, ensure doctors can prescribe a drug with confidence, protect patients. AMD is an eye condition that can result in poor vision. Doctors treat approximately 40,000 new cases of AMD each year. 2 drugs were licensed to treat AMD, drug A and drug B. In many other countries, drug C is also used to treat AMD, but drug C is only licensed in the UK to treat cancer. The cost per injection for each drug is very expensive for A, super expensive for B, cheap for C. The number of injections required to treat AMD is the same for each drug. In 2018, the High Court in the UK gave permission for drug C to be used to treat AMD. Evaluate the decision to allow the use of drug C to treat AMD in the UK. That means provide pros and cons. I want you to write this in full sentences in the exam, but I want you to see how I've arrived at my answer here, so I'm going to list it like this, just so you can see how we are working this out. So the main thing is drug C is cheaper. Let's work it out. It's 3 lots of £28, so it's only £84 compared with drug B, which was 3 lots of £800, which is £2,400, which is crazy expensive. Why is that a good thing? Well, it saves money for the National Health Service, the NHS, meaning that more people can be treated. What about how good it is as a drug? Well, it says that in other countries they've been successfully using drug C, so it means that it's likely to be effective at treating AMD. What about the cons then? Drug C has not been tested in the UK. Remember, it's been tested to treat cancer, but we haven't looked at it from an AMD point of view, so we can't be sure that it's safe to use and that it doesn't have any awful unknown side effects. And this is always a mark when we're looking at drugs to do with this AQA exam board. If you just say, yes, it's fine to use it, then you're setting a precedent for other drugs to not be fully tested or fully licensed or pre-approved in the future. So accepting drug C's use. May set a precedent for allowing other fully tested drugs in the future. So when you're asked to evaluate, please don't muddle up your answer. Just give a list of pros or cons. That's what the word evaluate means. I hope you found this video helpful, guys. Make sure you like and subscribe and comment if you want to see similar videos.